Today, we are diving into a technology that is literally rewriting the future of medicine. It's called gene editing, and it's a field that is moving at, well, just an incredible speed, promising to fix the very source code of human disease.
You know, at its heart, the goal is actually pretty simple. Imagine your genetic code is a giant book made of billions of letters. A single typo, just one letter out of place, can cause a devastating disease. So the big question is, what if we could go in and correct that one specific letter? That's the challenge, and the story of how scientists are tackling it is it's absolutely remarkable.
So our story really begins with the first huge breakthrough. It's a tool you've probably heard of called CRISPR, which, for the very first time, gave scientists a relatively easy way to cut and paste our DNA.
CRISPR-Cas9 is basically a programmable homing missile for our own DNA. Think of it like this. You have a guide RNA that zeroes in on the exact genetic sequence you want to change, and it brings along this protein, Cas9, which acts like a tiny pair of molecular scissors, just snipping the DNA right at that spot.
But here's the thing. While cutting the DNA is incredibly powerful, it's also a bit of a blunt instrument. When you completely snap both strands of that DNA helix, you're basically sounding an alarm bell, and the cell's emergency repair crew rushes in. The problem is, this repair process can be, well, it can be pretty sloppy. It might accidentally add or delete a few DNA letters, these are called indels, and you could end up causing brand new problems while trying to fix the original one. Not ideal.
So this whole problem with the molecular scissors, it really pushed scientists to ask a new question. What if we didn't have to cut the DNA at all? What if we could just erase the typo and pencil in the correct letter? And that line of thinking, that's what led to the next generation of gene editors.
And right here, you can really see the huge leap forward. Instead of that brute force cut from the scissors, this new tool is so much more like a pencil. It completely avoids breaking the DNA's backbone. It just chemically rewrites a single letter, which gives you so much more precision for fixing those simple genetic typos. And this new tool, it's called base editing. It's really clever. It still uses a modified CRISPR system to find the right address in the genome. But instead of cutting, it brings along an enzyme that performs a direct chemical change on a single DNA base, just one letter, flipping it to another one. No double-strand break, no messy repair crew, just clean, precise work. But, okay, this incredible precision comes with one major limitation. Base editors are specialists, they're not generalists. One type can only turn a C into a T, and another can only turn an A into a G, and that's it. Now, these two changes actually cover a surprising number of genetic diseases, but it's still a very, very limited toolkit. You just can't make all the corrections that you need to.
So the hunt was on for a tool that had that amazing targeting ability of CRISPR and the precision of base editing, but with the versatility to make any kind of change you wanted. And this is where prime editing enters the story.
All right, if CRISPR is the scissors and base editing is the pencil, then prime editing, well, think of it as the genetic equivalent of the find and replace function in your word processor. It can find a specific sequence and just rewrite it with unbelievable precision and flexibility. So how on earth does it work? Well, prime editing pairs a modified Cas9 protein with another enzyme called reverse transcriptase, which is basically a molecular copy machine. This incredible combo doesn't just change one letter. It can directly write a new corrected strand of DNA using a template that scientists actually give it. It's wild. Let's just walk through it real quick. First, the editor finds the exact spot in the DNA. Second, instead of a full dangerous cut, it just makes a gentle nick on only one of the strands. Then third, a special guide RNA, called a pegRNA, comes in carrying the correct genetic template. The reverse transcriptase enzyme uses that template to write the corrected code right at the nick. And finally, the cell's own repair machinery comes in and finishes the job, seamlessly weaving in the brand new sequence. And this is what makes prime editing such a total game changer. It just blows past the limits of the older tools. It can perform all 12 possible single-letter swaps. And not just that, it can also precisely insert or delete small sections of DNA. This amazing versatility makes it powerful enough to potentially correct the vast majority of known genetic errors that cause disease. It's just incredible.
Okay, so this all sounds amazing in a lab, right? But how does it move from a scientific curiosity to a life-changing therapy for actual people? Well, these tools aren't just theoretical anymore. They're already being tested in clinical trials with real patients right now.
We are seeing unbelievable progress. I mean, look at this. Clinical trials are already using these technologies to treat devastating blood disorders like sickle cell disease and beta thalassemia. In other trials, for diseases like hereditary amyloidosis, the therapy is being delivered directly into the patient's body to just switch off the faulty gene causing all the problems.
To really understand the potential here, I just want you to focus on this one number for a second: 87%.
So what does that 87% actually mean? It means that in a landmark clinical trial for a devastating liver disease, a single dose, just one shot of a CRISPR therapy delivered directly into the patient's bodies reduced the toxic protein that was causing the disease by an average of 87%. It is a stunning, breathtaking result that proves that gene editing inside the human body is not science fiction. It's a reality.
And the applications, they go so far beyond inherited diseases. I mean, scientists are using CRISPR to engineer superpowered immune cells, they're called CAR T cells, to better hunt down and destroy cancer. It's being used to create lightning-fast diagnostic tests. And by using tools like prime editing, researchers can now build incredibly accurate animal models of human diseases, like Tay-Sachs, which is going to speed up the search for cures exponentially.
So think about where we've come from. In just a few short years, we've gone from molecular scissors to a precise pencil to now a full-blown word processor for our own genome. The technology is moving faster than anyone could have possibly imagined, which leaves all of us with a really profound question. Now that we have the power to rewrite the very code of life, what should we choose to write next?